Extending the legacy of a classic reference, EcoSal Plus joins the ASM family of periodicals this June. This growing collection of continually published reviews reveals our ever-expanding understanding of the biology of E. coli, Salmonella, and the other Enterobacteriaceae. From cell architecture and growth to pathogenesis of infections to life in communities, EcoSal Plus is the single source of knowledge about these organisms and their use as model microbes. A distinguished editorial board, led by Editor-in-Chief James Caper, ensures that content serves the needs of the research community. Free trials available for institutions. Take a look at ecosalplus.asmblog.org.
Good evening, everyone. Thank you so much for coming out on this kind of gloomy evening to learn about West Nile virus, which I at least think of as a summer thing, a summer event, and it doesn't feel very summery tonight, but you all are going to be prepared. You will have heard all about it. I'm very, very happy to welcome Lyle Peterson and Roberta DiBiase to the ASM headquarters tonight to tell us a little bit about West Nile virus. I'm sure, let's see, I think all of you are old enough to remember 2003. Yes? Okay. Just checking. When you get my age, you're like, were you born then? Yeah. When West Nile first came to when it really started to get a lot of attention, 2002, 2003, and then it sort of disappeared for a while, at least as far as most of us were concerned, and we weren't really thinking about it. And then last year, suddenly it was in the news again. So what we're going to hear about tonight is what was up with that? What is it about the virus that gives you a pattern like that? Lyle's going to give us some information about that, how the ecology of this virus contributes to how it behaves and what we see. And then Roberta will tell us a bit about what the implications are clinically of this virus. So let me first introduce Lyle. He is the director of the Division of Vector-Borne Diseases at the CDC out in Fort Collins, Colorado. He has been in that position since 2004. He's an MD, but he also has a number of other degrees and training in epidemiology and public health. So he has the perfect background to be the guy who's watching this disease for us. His research now focuses on the epidemiology of these arboviruses, and by the end of tonight you'll know what that is if you don't now, and bacterial vector-borne zoonoses. So Lyle, come on up. Thank you so much for doing this, coming out from Colorado to speak to us.
And how do I steer here? Let's see.
I think it's
perfect. Okay, well, thank you for coming to this talk, my favorite subject. And so what I'll do is I'll start out talking about let's see if I can run this thing. How do you do this?
Left, right arrows?
I tried that. Nothing's happening.
That's worked for me. Huh, how about down?
Use the mouse and bring it to the bottom left corner, and then the arrows will pop up and press an arrow, and then you should be able to use the left right buttons.
Did you see that?
Okay,
It's right there.
got it. Okay, so what I'll first talk a little bit is give you a little bit of an introduction to the West Nile virus very briefly, and then talk about the epidemiology and the transmission of the virus, and talk about what happened last year in Texas and elsewhere. And then I'll turn it over to Roberta, who would talk about a little bit more in detail about the clinical aspects and more of the virology. So West Nile virus is a single-stranded RNA of a Flavivirus, Flavi meaning yellow. It's related to the yellow fever virus. And it's part of the Japanese encephalitis serocomplex of viruses, which includes St. Louis encephalitis virus, which is a related flavivirus that has been endemic in the United States, at least since the 1930s when it was first studied. The virus was first isolated in 1937 in Uganda in the West Nile district of Uganda, hence the name West Nile virus. And it has a very wide geographic distribution in the Old World. It extends throughout most of Africa, into southern Europe, throughout the Middle East, and Australia, but the one place it doesn't exist is in Southeast Asia, and nobody's quite sure why. And it was not present anywhere in the New World until it was discovered in 1999 during an outbreak of encephalitis in New York City, which was accompanied by a very marked epizootic, which resulted in thousands and tens of thousands of bird deaths. And the means of importation of the virus is unknown. There's a lot of theories about how it got there. This is one of them. And believe me, when it was actually true, people were really worried that some terrorist brought in the virus. I like this slide because it talks a lot about the British press. Like dog makes $60 million as a model. So the way it's transmitted is it's transmitted primarily by Culex species mosquitoes. And the virus overwinters in adult female hibernating mosquitoes in temperate climates. So what basically happens is everybody wonders, where do all these mosquitoes go? And in a northern place like Washington, D.C., or Maine, or Chicago, or places where you don't see any mosquitoes, basically these kinds of mosquitoes, what they do is the female goes into this thing called diapause, which is basically a hibernating state, and she finds this nice warm place to hang out. So your favorite sewer system or your basement, these mosquitoes just sit there on the wall all winter long. And then they emerge in the spring when it warms up. And so the virus is actually in the mosquitoes, and it's probably transmitted to those hibernating mosquitoes through transmission from transovarial transmission through the eggs. And so these mosquitoes that are infected are just sitting there in your basement or wherever, and they come out in the spring when it gets warmer, and they start a transmission cycle. And these species of Culex mosquitoes that we're concerned about are primarily bite birds. And so birds, unfortunately, are really good hosts for the virus. And many passerine species, like robins, for example, develop enormous levels of virus, like 10 to the 7th plaque-forming units per milliliter. You can find birds like crows, which get like 11 logs of virus. And some of these birds fly around happily with 11 logs of virus in their blood. So you basically got bags of virus flying around. And so these mosquitoes bite birds. So she comes out in the spring, finds a good bird. And American robins seem to be very good for this. Then in turn, more mosquitoes feed on that bird after it's become viremic, and which in turn infects more birds, as those mosquitoes bite more birds and infect them. And then finally, about the middle of the summer, you get huge numbers of infected mosquitoes. And you get so many infected mosquitoes that humans and horses become at risk. Humans and horses are actually not the favorite targets of these mosquitoes, but there are so many around that you actually, some of them will bite humans, obviously, and then humans become a risk. The thing is humans and horses do not create very high levels of virus in their blood. Although we get sick, the levels of virus in our blood is quite low. And so we actually don't infect mosquitoes very well, so we're considered dead-end hosts. So this is primarily a mosquito-bird thing, and humans are just incidental or dead-end hosts. So these are the three species of mosquitoes that primarily spread the virus. There's been more than 60 species of mosquitoes in the US that have been known to become infected with the virus, but these three are by far the most important. Culex pipiens in the north, which is appropriately named the northern house mosquito, Culex quinque fasciatus in the south, or the southern house mosquito, and Culex tarsalis in the west, which happens to be a very, very good vector, and we'll explain what happens with that later. So I'm going to just touch briefly on the clinical presentation, just to provide a little bit of background for what I'm going to describe with the epidemiology. And then Roberta will talk a little bit more, a lot more in detail about the clinical aspects. So if a person gets infected with a virus, about 70 to 80% of the people will have no symptoms at all. About 25% will develop what's called West Nile fever, which is kind of a misnomer because about half the people don't have fever, but they still get sick. And less than 1% will develop what we call West Nile virus neuroinvasive disease, which is meningitis, encephalitis, or acute flaccid paralysis. And about 10% of the people who develop neuroinvasive disease will succumb from the infection. Now, the important part about this is that obviously we have difficulty measuring asymptomatic people except through blood screening. People with West Nile fever, less than 1% of those people actually are diagnosed, go to a doctor and are diagnosed. And so the thing that we really know a lot about is West Nile neuroinvasive disease. And because most of those people end out seeing a doctor or diagnosed, end out in the hospital and are reported. So when I talk about the epidemiology in the next batches of slides, I'm going to focus on neuroinvasive disease for that reason. So this is what happened. I'm going to show you a series of maps. And this map here shows the situation in 1999. And you really can't see much going on. And the colored dots are the dots of the incidence of human neuroinvasive disease. And the green area is area where we detected the virus, mostly in dead birds. And you can't see much going on here because it was very focal in 99. There only human cases occurred in the New York City area, although the virus was found throughout New Jersey, parts of New York, Connecticut, as far as Baltimore. So the virus had spread much further than the human cases. And this is what happened in 2000. The virus went north in the beginning part of the year, up in the upstate New York and Massachusetts. And then in the south, although you can't see it because it's pretty small here, but we could detect virus kind of going south with the southern bird migration towards Florida. And so in 2001, there was green areas you could see that extended pretty much past Chicago, throughout the eastern part of the U.S., and human cases started appearing in the Florida and the southern states, as well as in the northeastern states as well. And then in 2002, the whole situation changed, where we had the first massive outbreak. And you can see that the virus had spread all the way to Colorado by that point, but caused this huge outbreak in the Chicago area, Louisiana, Mississippi, parts of Texas. And in 2003, there was another massive epidemic in the Western Plains states. And unfortunately, I became one of those dots because we had a big epidemic in my town. And the virus had spread all the way to the West Coast. And in 2004, we sort of started entering this endemic pattern where you just saw cases scattered throughout the U.S., but mainly in the midsection of the country, in California and elsewhere. So this kind of pattern continued in 2005, 2006, 2007, 2008, 2009. In 2010 and 11 look basically the same. I'm not going to show those, but you can see that the virus became endemic throughout the U.S., and there were sporadic cases all over the country every year. So this is the situation as of 2012. And we had 37,000 cases reported to us. 16,000 are neuroinvasive disease, resulting in about 1,500 deaths so far. The number of non-neuroinvasive disease, 20,000, is really quite an underestimate because as I explained earlier, that non-neuroinvasive disease cases are grossly underreported. So if you make an extrapolation of these 16,203 neuroinvasive disease cases, there's about 30 to 70 West Nile fever cases for every neuroinvasive disease case. So we estimate that there's been somewhere between about 500,000 and 1 million fever cases, West Nile fever cases, that actually has occurred so far, and somewhere between 2.4 and 4.9 million infections. So it's still, it seems like a lot of infections, but really that's only about 1% of the US population. So the vast majority of the population is still at risk. So this is the incidence of neuroinvasive disease by year, and you can see from 99 through 2001, not too much happened. In 2002 and 2003, there was those two big epidemics, which you saw. And then it kind of resulted in this endemic pattern with kind of a decreasing incidence through about 2011, which unfortunately paralleled my division's budget because people were no longer concerned about this until 2012 when it came back. And we'll talk about why it came back later. Now, as I mentioned about this cycle, it kind of revs up through the spring. So although the weather is kind of crummy right now, the cycle is just starting to rev up right now. And then about week 34 to 37, these are CDC weeks, and it corresponds to about the middle of July to the middle of August is when it usually peaks. And as you can see, these outbreaks occur rather rapidly because it is a stochastic process. These are some of the data of clinical cases we've detected through our surveillance system. And as you can see that there's slightly more males than females reported, except for encephalitis and meningitis. And it seems in AFP, acute flaccid paralysis, where it appears that males seem to be at somewhat higher risk than females for developing severe disease. The age that people are, encephalitis, people are generally older, but people with fever, meningitis, and acute flaccid paralysis, the median age is somewhere around 50 years of age. And you can see from this chart that 23% of the fever cases were hospitalized. It's probably artifact, since most fever cases probably never get recognized. But you can see that of the meningitis and encephalitis cases, most of them end out in the hospital. And you can see that pretty much all the deaths occur in people who have developed encephalitis or acute flaccid paralysis. Now this is the incidence of neuroinvasive disease by age group. And what's interesting about this is that we know that of people who get infected with the virus, it's about the same in every age group. Mosquitoes don't care how old you are. And we know this from blood donor screening where we can detect virus in people. And we find that the age, your probability of getting infected does not change by age. But your probability of getting neuroinvasive disease increases rather markedly. And we make the recommendation that people over 50 years of age, are higher risk for getting severe disease. But if you look at this, you're really going downhill from birth. Your risk about doubles for every decade of life. So there's maybe some philosophical thing about that. We can talk about that later. But what's interesting about this is if you divide up these neuroinvasive disease cases, and you saw that big increase going up as you get older, that's primarily almost always due to encephalitis. You can see that the incidence of encephalitis really markedly goes up the older you are. And in fact, by the time you're 65 years of age or greater, if you do become infected, you have about a 1 in 50 chance of getting neuroinvasive disease compared to about 1 in 2,000 for somebody in their younger 20s. Now, if you add up all the cases that have occurred from 1999 through 2012, and these are neuroinvasive disease cases, you can see that certain parts of the country appear to be consistently higher risk than other parts. There's a real hot spot in the Plains states, up around Chicago, Detroit, the upper part of the country there, and another big hot spot down Louisiana, Mississippi. And part of the reason, we don't really know why this occurs, but one of the reasons that we surmise that this big black zone there, at least the one in the western plains states, is that Culex tarsalis likes to breed in irrigated farmland, and it's a very good vector for West Nile. There's a lot of irrigated farmland in that area, which may account for the reason we see some of this pattern. But for the most part, we really don't understand totally why this pattern actually occurs throughout the country. But if you look at the number of neuroinvasive disease cases by county, you see kind of a different picture. You know, back here, there's this big black zone in North Dakota and South Dakota, but until the oil boom, nobody ever lived there. And so what you see is places like Dallas and Chicago and Detroit and northern Colorado, Denver area. You see those are really, they have a lot of cases because those are where high population centers intersect with this area of high incidence. So it produces a lot of cases. Now the thing about West Nile virus is it's very sporadic. We really don't understand why outbreaks occur in one area versus another. And on this slide I've listed the proportion of US counties reporting one or more cases of West Nile virus disease from 2002 through 2012. Turns out that 19% of US counties will report one or more West Nile virus. An average of 19% of the counties will report one or more West Nile cases. So the vast majority, 4/5, in any given year will not report any West Nile at all. But this ranges from 8 to 35%. And overall, 67%, or 2/3 of all US counties, have had at least one case of West Nile virus neuroinvasive disease. And this is the West Nile virus disease incidence in 2003, 2006, 2010, and 2012. So what you can see, remember that map I showed you cumulatively what happened. But if you look at any given year, what you see, and what makes this disease so fascinating is that there's counties that have very high incidence. The very next county next door has no incidence, and it may reverse itself in a subsequent year. So the diseases, or the incidence is very, very focal and from one place to another, which makes it very hard to predict where an outbreak is going to occur in one year or another. And at the bottom right, you can see what happened in 2012, where there was this massive outbreak in Texas, Louisiana, Mississippi, in the upper plains states, around Chicago, Detroit areas, and also quite a few cases in California. So make a mental note of that map. So why did this big increase occur in 2012? Well, the kind of the smoking gun was it was really hot last year. And on this map, I have the ranking from 1 to 118 of the hot, over 118 years of recording, each state's rank of where they were. So the state that ranked 118, which is the record hot, was the hottest year on record for the last 118 years. And you can see from this map that almost every state was way abnormally hot. We had the 4th warmest winter, a record warm spring, the 2nd warmest summer, and a warmer than average autumn. So why is temperature important? Temperature does a couple of really bad things to these arboviral diseases. One is it shortens the time from when a mosquito becomes infected, so when it bites, let's say a bird, and gets infected, to the time when it gets infectious. So what happens in the mosquito, the virus bites a bird, it ingests the virus has to pass and multiply in the mosquito, pass out of the gut, multiply in the hemolymph of the mosquito, migrate to the salivary glands, and reach a high enough concentration in the salivary glands, and hence the saliva, so that when it bites you, it can transmit the virus. That process takes a number of days. That process is called the extrinsic incubation period. So if you increase the temperature, that period shortens. The other thing that temperature does is it increases the viral replication of the mosquito. So when it's warmer, the mosquito develops much higher concentration of virus in its blood and is able to more easily transmit infection. So here's what happens when temperature is cool. The virus, or the mosquito, emerges as an adult. It mates, and we can talk about mosquito mating later. It's kind of an interesting process. But so the mosquito mates, and the female mosquito is the only one that takes a blood meal because it wants that blood to make protein to make her eggs. So the mosquito will feed on the infected bird, become infectious itself. So at the beginning of the yellow bars, that's when she gets infected. She goes out and lays her eggs, and then she may feed on a person. If that extrinsic incubation period is long, in other words, in cool weather, there's not enough virus yet in the saliva of that mosquito to infect a person. So in cool weather, the mosquito has to live a really long time to pass that extrinsic incubation period to be able to pass the virus. But if it's warm, what happens is that extrinsic incubation period is short, so after she lays her eggs and then goes to take another blood meal, she's infectious, and that's when you get infected. So temperature is very, very critical to this whole process. The other thing that's important is rainfall. Obviously on the left or the right are not conditions suitable for breeding mosquitoes. And so the optimal breeding condition is somewhere between a drought and a excessive rainfall situation. And if you look what happened here last year, much of the US was drier than average, and this is a similar map to what you saw before. This is drought. You can see kind of the states in the middle there had exceptionally high, strong drought conditions. But in Texas, for example, and those northern plain states in Chicago and elsewhere, where we saw these big outbreaks, there was actually a slight drought or fairly normal rainfall. So what you had is a combination of very high temperature with a fairly normal rainfall. And that seemed to have set this whole thing off. And you can see better on this slide where you can see the drought on the right map and the cases of neuroinvasive disease on the left map. You can see where it was really, really dry, not a lot of West Nile, but it was in that perimeter area around that drought zone where we saw a very high incidence. So it was a very high temperature with fairly normal rainfall. So in summary, we can say that West Nile virus is an important cause of neurological disease in the US. It's by far the most common cause of arboviral encephalitis in the US. We have no vaccines or therapeutics for this virus, and there's a variety of reasons for that. One of them is trying to figure out how to bring them to licensure. How do you do a phase 3 clinical trial when the cases are scattered all over the place and unpredictably so? The other thing is that seasonal outbreaks occur annually but are focal and unpredictable in size or location. And certainly we need to have consistent and adequate long-term support for surveillance and response. It was like what happened in Dallas. They didn't have any West Nile virus for several years through that long period where nothing much was happening, and so they fired their entomologists. The mosquito control capacity went down, and the ability to respond went down just as the time the whole thing resurged. So that's it, and I'll turn it over to Roberta. Thank you.
So if you don't mind, we'll wait and take questions for both after both talks. So I'll introduce Roberta DiBiase. She's an associate professor of pediatrics at the George Washington School of Medicine, as well as being the acting chief and attending physician in the Division of Pediatric Infectious Diseases at Children's Hospital here in Washington. She's also a principal investigator, and she works on improving treatments for viral encephalitis. I can say the plural of that word. As well as influenza, herpes simplex virus, congenital cytomegalovirus, and adenovirus. So lots of viruses. And she also treats immunocompetent and immunocompromised patients at Children's Hospital. And she's going to talk to us a little bit about what this West Nile virus disease looks like from the point of view of the clinician.
Thanks for having me. This is also one of my favorite topics. So Lyle has given us a lot of information about the transmission and the birds and the mosquitoes, and I'm going to talk to you now about you, the human being, and what this does. And just to give you a little preface, encephalitis, or infection of the brain, is one of the most frustrating diseases to diagnose. It's one of the most frustrating diagnoses to give to a patient because we don't have treatments. So it's for this reason that there's really a lot of research going on in this area, and why I remain involved in the area. So West Nile virus is a single-stranded positive sense RNA virus, meaning it can immediately come into the cell and act as a template for it to start making more of itself. And it turns out that all strains, human, mosquito, and bird, are greater than 99.8% identical in the U.S., and they are very closely related to the Middle Eastern strains that we knew were circulating in the 60s, 70s, and 80s there. But what was different when we saw this explosion in 1999 and moving across the country across the subsequent 10 years is that there was more neurovirulence than had been appreciated when it was endemic in Africa and when we had these cases in Israel. So there was more viremia and mortality in birds, and there was certainly higher frequency of neurologic disease appreciated in humans. And we don't really have a full grasp of why that is, but there's a lot of research to look at that. And some of the things that have been raised are perhaps glycosylation of the envelope protein of the virus, because that envelope protein is very important for entry of the virus, and also non-structural proteins. So NS4B in mice, if that is mutated and put into a mouse, they actually have less neuroinvasive disease. So lots of research going into why it seems now more neurovirulent than it used to be. So let me tell you a little bit about the clinical disease, and we purposely put this in twice just to reorient you. So just to reiterate, what we're talking about for neuroinvasive disease is the very, very tip of this iceberg, and even a smaller percentage are fatal. But I guarantee you, when you see a child or an adult come into the hospital with severe disease, and I'll give you an example of a case we recently took care of, it doesn't matter that you're 1 out of 150. It's you that is paralyzed or has seizures and is going to have long-term sequelae. So the pathophysiology of the disease is we have the bite from the mosquito. There's local replication of the virus in the local lymph node drainage of where you were bitten. And then there's replication that leads to a secondary viremia in our reticuloendothelial system, so like our spleen and other immunologic organs. And then there's a peak viremia where if you actually knew you were going to get sick and you measured virus, there's a peak viremia about 2 days before you actually present with your illness. And this was an important thing to recognize, because when patients actually come in with clinical illness, 2 to 14 days after they were bitten, we often can't find the virus by PCR, and we have to rely on serologic methods. We don't have time to talk about it, but what was also appreciated when it broke out into the U.S. is that new modes of transmission of this virus were appreciated here. So we found that patients that had organs transplanted directly, unfortunately from a donor who we didn't know had West Nile virus, of course it's going to be much shorter than 2 to 14 days before those patients got sick. So very high levels of viremia directly inoculated into a patient and bypassing this whole primary and secondary viremia. So what do these patients look like? As Lyle mentioned, West Nile fever is the vast majority of patients, and he's right, we rarely see these patients come into the hospital at all. They do come into primary care physicians' offices or into our emergency room, and it's a very nonspecific febrile illness. So there's really nothing about it that would distinguish it from any other febrile illness that we see in children or adults. They generally have fever. They'll have headaches, sometimes body aches. There is sometimes some GI symptoms, so they'll come with nausea and vomiting. It can be mistaken for a routine viral gastroenteritis. About half of the patients can have a rash, which is very nonspecific. They can have swollen lymph glands, and they usually feel very tired, even after their rash recedes. And Lyle's nodding his head, so I think when he had it, he felt tired too for a while. But what we're really going to concentrate on then is that tip of the iceberg, that 1%. And you know, this disease has been around a long time, and in Africa and Israel, it was well appreciated that the disease caused meningitis and encephalitis. And just to clarify that for people that are not medically inclined here, meningitis is inflammation of the lining around your brain and your spinal cord, and encephalitis is actual infection of the brain itself or the spinal cord. And there are differences in how those patients present to us. But what was really more new and less appreciated when it had been circulating in Africa and Israel, but what we realized here in the U.S., particularly during that 2002 and 2003 year, were other presentations such as acute flaccid paralysis, which for all the world looked like polio. And I know none of these people in this room are old enough to have seen polio. I actually have never seen a natural case of polio, but my colleagues who took care of polio patients said it's absolutely identical. So patients come in with paralysis of one or more limbs. And it was also appreciated with whether or not they had encephalitis or AFP, that they had movement disorders. So jerky activity, cogwheel rigidity, if you're familiar with Parkinson's disease, because of the areas of the brain where this virus likes to go, a lot of the patients have those kind of symptoms, which is very difficult for rehabilitation and your activities of daily living if you can't control your motions. And then a whole host of other things have been associated with West Nile that were relatively new. But what was interesting, I was actually in Colorado in 2002 and 2003, and I have a lot of adult neurologic colleagues, and my mentor was an adult neurologist, and I had the opportunity to see lots of adult patients, but I was the primary consultant for pediatric patients, and I kept saying to myself, where the heck are the pediatric cases? So we kind of recognized right away that there must be some difference. We actually sent out surveys to all the pediatricians in Colorado to try to identify cases, prompt them. And there really weren't that many cases. So it's very clear, even at the beginning of this epidemic, that there was a huge difference in age. But having said that, we did have cases. It's not that there weren't any. So the incidence of neuroinvasive disease increases with age, and in children was only 4% of all neuroinvasive disease. But when we saw it, it was usually aseptic meningitis, so the kids usually got better, less so than the encephalitis and the acute flaccid paralysis. But we did see these disturbing cases every now and then, and we just recently had this patient, maybe last summer, I believe. So this was a previously well 16-year-old boy. He is not only an average 16-year-old soccer player, but an elite soccer player. He was going for a college scholarship. He's, plays on other travel teams, and he presented with bilateral lower extremity weakness. He thought about it for a while, and he had had a very aggressive soccer tryout and kind of had attributed feeling crummy to this tryout, but had fever, headache, and neck pain 5 days before this weakness, and it rapidly progressed. Over 2 days, he actually lost the ability to stand. He had bowel bladder dysfunction, meaning he couldn't control his bowel or urination, and he came into Children's Hospital, had imaging, which I'll show you a typical picture of what this looks like, but he had definite abnormalities consistent with this poliomyelitis syndrome. We did a spinal tap, and he had inflammation of the spinal fluid, which is very characteristic, and indeed his serologic testing confirmed that he had West Nile virus, both from his spinal fluid as well as his blood. So this AFP syndrome, as Lyle's alluded to, and what we definitely saw in the adults, is it occurs in this younger population. And when I correlate this to the adult patients I think I saw 10 patients in Colorado during 2003 in about a 3-month period where they were healthy 40 to 50-year-old women or men going to the gym, taking care of their kids, doing all the things that 40 and 50-year-old people do who are active, and then the next day they're paralyzed. And it's very shocking and disconcerting to see this. And we saw the similar thing in this teenage boy. This is a picture on the right that shows some destruction of the anterior horn spinal cord motor neurons, which is a typical pattern for what you would see in either West Nile or poliovirus-induced poliomyelitis. And they tend to have painless weakness, but nevertheless, if you can't stand, that's bad, even if it doesn't hurt. And we talked about the CSF findings. This is a typical MRI, and if you're not used to looking at MRIs, it's essentially like taking a slice of bread through your body. You're taking cuts this way, and this right here is at the level of your spinal cord, and this white area should not be here. So this is inflammation and destruction of the anterior horn of the neurons that would lead to that presentation where you have the weakness and paralysis. In our patients that don't have this AFP syndrome but who have standard encephalitis, it's very routine to get an MRI scan to look for actual whitish spots essentially in various parts of the brain. And it turns out in West Nile virus, even when we confirm it, we often don't see any abnormalities on the MRI. But what has been appreciated is if you see an abnormality, it tends to like to go to certain areas like the thalamus and also the midbrain. And these are, for instance, the midbrain is highly correlated with Parkinsonian type symptoms. So it kind of explains what we see from the clinical standpoint. And then what happens to these patients? You can certainly die, particularly if you're older. Up to 35% of these elderly patients with encephalitis can die. But even if you don't die, a large percent of these patients have significant impairment, even a year after their diagnosis. And I think one of the interesting dichotomies is if you have encephalitis, you can be really sick, be in the intensive care unit, have intractable seizures. But if you survive, you have a better likelihood actually of recovering your baseline function as opposed to someone with this poliomyelitis syndrome. I was really impressed by the patients I saw in Colorado that if they came in very impaired and actually had abnormalities on their MRI, the vast majority of them did not recover a significant amount of function. So we were very worried for this boy who came in, who was this elite athlete, to see what would happen. But I'll give you the follow-up on him, which is actually a happier story than that. Even if you recover your motor function, a lot of these patients, and particularly in the subsequent years in which they've been studied over a long period, have cognitive impairment with their memory, difficulty concentrating. They can have disabling fatigue where they can't go back to work or go back to school. We've already talked about tremor and Parkinsonism. And then more recently appreciated is our neuropsychiatric symptoms. So a large percent of these patients, and understandably so, have significant clinical depression because of their impairments over this long period of time. And is extremely costly from a rehabilitation standpoint. So our patient was treated with intravenous immune globulin, which I'll talk about in a moment as a possible treatment. He was actually wheelchair bound and could only go with a walker at the time he was discharged, about a week and a half after he was admitted. He went to National Rehab Center and lived there for 4 months doing extensive physical rehabilitation daily. And made some gains, and then he continued as an outpatient with more daily outpatient therapy. And you can imagine, of course, he couldn't go to school at that point, was getting homeschooling. But I'm happy to say 9 months later, he could independently ambulate. He does still have some remaining deficits in his left lower leg, so he can't cock his ankle completely normally, but compared to how he was, we're extremely happy with how he did. He has no cognitive deficits whatsoever, and he is able to participate in his, club level sports. But his soccer career as a college, athlete is really over. So this has serious implications for him. But obviously, we're very happy with how he did compared to some of the patients we see with AFP. So how do we diagnose it? I'm not going to spend a lot of time on this. I think the most important point and why I boxed it is that a lot of times we think of PCR, and I'm sure you've talked about this at some other lectures, that how sensitive it is and how great it is at picking up viruses that we'll miss by other technologies such as culture. But for the reasons we talked about the kinetics of the disease, by the time they're coming to see us, they are generally PCR negative, unless they have an immune compromising problem or a transplant patient. So we really do rely on serology, and I have a slide here to just show you that. So you can see the peak viremia is going to be 2 or 3 days before they present with their clinical symptoms shown here. And about 4 to 6 days after their clinical illness, we can start detecting the IgM, both in the CSF and the serum. What's interesting about the IgM response is that it lasts a very long time, which is a little different than what we're used to for IgM serology for other illnesses. It's usually kind of a quick up and down. And this can actually screw us up a little when we're trying to determine if the patient in front of us really has West Nile virus or they actually had West Nile virus a long time ago. And then later, about 2 to 3 weeks after illness, they'll generate late onset antibody, IgG antibody. This will persist lifelong and is protective. So, after taking in all this data, the question that we often ask ourselves and that our patient's going to ask us is, why did this happen to me? Why am I the 1% of the iceberg? What is it about me that caused this to happen? And so people that study this area are interested in both host factors and factors that are involved with the virus itself. So some people concentrate on the virus, some people concentrate on the host. And these are some of the host factors that have been appreciated to predict that if you have West Nile, that you're going to have a higher likelihood and be in that top 1%. So our solid organ transplant patients, because you're putting an organ in that has West Nile directly in, that's going to be an extreme risk for invasive disease. Hematologic malignancies, so our leukemia, bone marrow transplant patients, anyone with any kind of impaired T cell function or who is receiving therapies for treatment of diseases that will decrease their B cell function, maybe hypertension and cardiovascular disease. I think that's a little less clear. But there's a whole body of research of which can't be summarized on one slide. I just wanted to give you a couple of ideas about what's going on in the research world about this that are potentially identified host factors that may set people up that we know are important for control of the virus. So there's innate immunity, meaning things that we have that are ready to fight off any infection or pathogen, not specific to the pathogen itself. So these are things like toll-like receptors and interferon, and these are indeed important for control of West Nile virus. We know that humoral immunity is important, so antibodies that you make after you've been exposed to the virus, and these are targeted against the envelope of the virus, which surrounds the virus, as well as some non-neutralizing antibodies to other non-structural elements of the virus. Cellular immunity, so these are T cells, regulatory cells, and cytokines. All of these are important to varying degrees for control of this virus. And the reason we want to understand these host factors is if we can get a handle on this, it helps us to develop targeted therapies, which we don't have right now. And then what about host genetics? So I'm sure you've been reading about and hearing about how, now that we can identify the human genome, we're going to be able to figure out everything that we need to know. So, West Nile virus is no exception to the rule where people have looked at the genome and tried to figure out if this can tell us anything. So there have actually been 2 human genes identified that seem to have an important role in West Nile virus disease. One is the OAS1 gene, which is involved in degradation of viral RNA, and this correlates with an increased risk for getting infected, but not the severity of the disease. Whereas the CCR5 chemokine, which you may have heard of before as a co-receptor for HIV infection, this actually leads to an increased susceptibility of severe infection. And in fact, 1% of the U.S. population is homozygous for this particular mutation. So that's 1% of the population that's theoretically at a higher risk of neuroinvasive disease. And then the converse of that is if we take patients who actually have severe neuroinvasive disease, 8% of them will have this homozygous mutation for CCR5. So treatment, it's terrible. We basically have to tell these patients, yes, we know all this interesting science, but we have nothing to help you right now. It's very frustrating to us and the patients. And this is another whole area where there's active research based on what we've learned in vitro, in animal models, and then now moving into human data. So ribavirin is a drug that we tend to pull out anytime we have nothing else and see if it works. And it turns out it's not likely to be a benefit for treatment of West Nile virus. It actually showed some detrimental effects in an animal model. And even though it wasn't the greatest study, probably, if anything, had no effect or made things worse in a human model. Interferon alpha actually probably has some potential benefit, and I have to say that's the one thing that I tend to use empirically in a patient with severe disease, and we did use it in this patient. Whether or not it made a difference, we don't know, because it wasn't a controlled experiment, which it never is when you're treating a patient. But it has shown in vitro efficacy. If we look at cells, it actually looks good in a couple of animal models, and there is a randomized controlled clinical trial in progress in humans for that. The hyperimmune West Nile virus, IVIG, is a product that, if you think about it, in a place like Israel where it had been circulating a long time, if you took samples of blood from patients, or from people, there's a higher incidence of seropositive people. And if you then pool all those patients who are donating blood, which is how we make IVIG, or pooled immune globulin, you're going to have a significant percentage of titer that is theoretically protective or could help people. And that was the product that we actually tried to study through the Collaborative Antiviral Study Group right after that huge peak of disease in 2002 and 2003. But unfortunately, as you saw from the graphs that Lyle saw, by the time we were ready to do this trial, there was hardly any disease. So unfortunately, it was underpowered. I've never seen results for that. I don't know what the numbers were, but right now we don't have data to know if that works or not. And then there are monoclonal antibodies that target that envelope protein that are very important for entry. We don't have any results yet from a phase 1/2 trial that was completed in that area. There are antisense oligomers. So antisense oligomers bind to the viral RNA and inhibit the replication of the virus. Phase 1 trials showed safety, but the large randomized trial that was planned was terminated because of poor enrollment, again, because of the incidence that went down so significantly. And the same goes for siRNA. So that leaves us with prevention. And there are things you can do as a person and things that can be done as a community to help prevent this disease. Lyle is obviously the guru of the community interventions, but these are things that you see here, such as spraying areas for mosquito, areas that have large amounts of mosquitoes that are infected, that are circulating. But as a person, what we tell our patients and what you should know is that you can use sprays such as DEET-containing sprays. The amount of protection you'll get depending on the percentage of DEET that you use is important to know. A lot of people don't know that. So if you're going to be outside for 5 to 8 hours and you're not going to have any other spray, you need to use a very high percentage of DEET, so 30 to 50% DEET. Whereas if you're going to go out for an hour, you can use a lower percentage. You need to look at your can and see if you're getting effective coverage, or you need to keep putting it on. Repairing screens, so you want to prevent mosquitoes from coming into your house. And in Colorado, no one has screens, so we used to always laugh when I showed the slide. And then you want to be very cognizant of any standing water in your yard. So even dog bowls and anything that collects water, you need to change those frequently because those are areas where mosquitoes breed. So the last slide is just about vaccine, and you know there have been many equine vaccines that are highly effective. There are human vaccines in clinical trials, and it's beyond the scope of this talk to talk about all of them. But there is a product by Sanofi Pasteur, and these are chimeric constructs where they take the backbone of a dengue or yellow fever virus and then put in West Nile virus antigens. So the one that's been studied in phase 1 and 2 trials in 18 to 65-year-old patients was found to be safe. There was a phase 2 or larger expanded study that was randomized, controlled, placebo-controlled, double-blinded, and looked at different doses in adults greater than 50 years of age. Again, was highly immunogenic, meaning they make protective antibody, and again, was well tolerated and safe, and this was recently published. So these studies are moving forward, and will remain to be seen if this becomes used in a widespread manner or not, but I think there's hope on the horizon for a vaccine. So this is just, there's great cartoons about West Nile virus. So this is the Homeland Defense, billion-dollar jet saying, "Don't ask me, we've been ordered to shoot it down." This is a little West Nile virus-infected mosquito, because you can imagine there's a lot of public health outcry when these outbreaks occur. So I'm going to end there, and then Lyle and I can take any questions you have.
So I should mention that our audience tonight is not only those of you in this room, but a vast crowd of people in the cloud watching these talks at ASM student chapters and elsewhere. And the video will also be archived, and it'll be hosted on the MicrobeWorld website. So if you want to see it again, or if you have friends who are interested in it, it will be there. I'm sorry to those of you watching this in the future. You won't be able to ask any questions, but hopefully somebody in this room or watching remotely will ask the question that you are thinking of right now. So we'll take some questions here, and we'll take some questions that come in over the internet. You had a question?
Yeah, I did have a question. Hi. So you had mentioned the increased incidence of neuroinvasive disease in these American cases. And I'm wondering if it could be, I mean, I'm sure this has been addressed already, but is it simply because it's a naive population as opposed to in the Middle East where it's been circulating for a long time and people might have been exposed?
I think the answer is no, but Lyle may know more about the actual changes in the virus.
Right. What happened was very interesting, and that was around the mid to early 1990s. There was a new clade of viruses that emerged, first in Africa, where this virus is obviously endemic, which seemed to have increased pathogenicity. And so it started causing outbreaks in northern Africa, and then subsequently in Europe. There was a huge outbreak in 1996 in Romania. And this clade of viruses that seemed to have increased pathogenicity started causing increased neuroinvasive disease in Europe, starting with this outbreak in Romania, subsequently in Russia and elsewhere, and persists to this day. And it was a virus that was among these clades of viruses that was actually imported into the U.S. And what's happened that we know is that there's been a couple of mutations in the virus which have been well studied, one of which causes increased mortality in birds. And it was that virus that has a single mutation in the NS3 helicase gene, which seems to kill birds for some reason. Another mutation happened in the virus, which was fascinating, because the original virus that was brought into the United States has completely disappeared and was replaced starting about in 2002 with a new virus that was the daughter of this virus that was introduced, which this virus that it subsequently took over has another mutation in it, which makes it very sensitive to temperature. And that may be one reason why we're experiencing these huge outbreaks with increased heat, with these heat waves that have occurred. But as far as the virulence of the virus in humans really don't have much to do with it. You know, it's really the virulence in birds that is the big factor, because that's the primary host. We're dead ends, so we don't matter to this whole cycle.
Thank you both very much. I'm always interested in the prevention side, and so maybe two questions. One would be, you know, if you think it is really temperature dependent, if you can use any sort of, weather models or whatever to try to, predict what the next season's, hotspots might be. But even if you could, when you have a lot of this virus in very rural areas, I mean, I realized last year Dallas was the hot zone, but in rural areas, spraying isn't probably going to work. And again, you can go back to the screens and the DEET and all that. But is there anything else we can do for prevention?
That's an excellent question, and one that we debate every day. And first, the temperature dependence. What you saw was a map of the U.S. where basically the whole country was abnormally hot, but these outbreaks, for whatever reason, only occurred in certain counties, or it was highly focal within the country. So temperature seems to set this off, but it's not very good at predicting exactly where the outbreak is going to occur. It just says there's going to be an increased probability of an outbreak, but whether it's going to be in Dallas or Austin or Los Angeles, who knows? And for that reason, temperature by itself is not a very good indicator of whether some place should do, whether we can target prevention efforts. If it's abnormally cold, it's not going to happen. If it's abnormally hot, it might happen, but we don't know where. The problem is, as you point out, in a rural area, it may have a high incidence, but you can't spray half of Kansas or whatever, so that becomes a problem. Really, control has to be fairly limited to metro areas, which have high populations and a relatively small geographic footprint. So I think the answer to this may be possibly development of better therapeutics to treat it, for one. But I think the issues about vaccination become important. Should we develop a vaccine for humans? That may be the only answer for some of these rural areas.
Shannon, do we have any questions from online?
Yeah, we do actually. So I had a question specifically a little bit more for Roberta. We had someone from Twitter who was really intrigued by what you were talking about with regards to the PCR testing and how once the patient starts to actually present, you often are getting PCR negative results as the immune response is kicking in. So our Twitter follower was wondering, is the immune response actually generating the symptoms that the patient is experiencing?
Yeah, that's a great question. We didn't go into a lot about this, but there's certainly a role for cytokines in the pathogenesis of the disease. And it's kind of interesting. In neuroinvasive disease, a cytokine response is actually going to be damaging to the brain. So the virus actually induces some mechanisms to decrease that cytokine response. But the downside of that is when you decrease the cytokine response, you're limiting your body's ability to clear this virus. So it's this dichotomy of inflammation versus non-inflammation. So I think definitely some of the symptoms we're seeing, seizures and inflammation of the brain, are due to some of that cytokine response. Definitely.
So I guess my question is probably mostly for Lyle here, but the question is, how uniform do you think our surveillance actually is? I can't help but notice that in the 2012 epidemic that you see these outbreaks around areas that study West Nile virus. So is the reason that we know there's an outbreak in Texas because Alan Barrett works in Texas? Or is it possible that there's sort of a more widespread that clinicians in somewhere that's not studying that or having that in the headline or not necessarily reporting that or testing for West Nile?
That's a very good question. I don't think it has to do with the distribution of West Nile researchers. But as I mentioned, the neuroinvasive disease is pretty obvious. I mean, there aren't that many people that come in with encephalitis or paralysis. And so these cases are mostly picked up. And I want to say mostly because even in places experiencing pretty good outbreaks, we find that only about 50 to 80% of the people with compatible disease are actually diagnosed with West Nile. But those cases are pretty well picked up throughout the country. We do have a national surveillance system called ArboNet, which every state health department participates in. And so these cases, we really make a big effort to go out and find, no matter where they are. So it really is a ubiquitous problem right now in the U.S. It's just worse in some areas than others.
I had two questions, actually. One is, thank you for the great talk. We heard about the humans and the vector, but is someone following the birds? So are you matching like orthogonal data from researchers on bird population throughout the U.S.? And the other intriguing thing for me was, why not Florida, which seems to have an elderly population, birds, and mosquitoes?
And it's hot.
And
it's
it's hot,
hot.
and there isn't an outbreak.
Right. So I think there's a few answers to your questions here. First, let's take the bird question. It's actually surprising how hard it is to follow birds. They do annual bird counts, and birds fly around, and it depends on the time of the year, and I'm not an expert in that. But people who are notice really marked reductions in bird populations of highly susceptible species like crows and jays and other birds that seem to die off pretty readily. But those populations are actually coming back. And one of the areas that we're very interested in studying is are these birds becoming, I don't want to say immune, but less susceptible to severe neurological disease than they were, let's say, 10 years ago. And we think that's probably happening, and there's a variety of reasons why the birds may be adapting. I mean, certainly there's a tremendous selection pressure. And if you look at birds in Europe, where the virus has been around for a long time, or in Africa, you don't see these bird deaths. So I think the birds are adapting. It's one of the areas of pretty intensive research now to really try and figure out the mechanisms for this adaptation. The second question is why Florida hasn't been particularly hard hit. Certainly Florida's hot, and so is the entire Southeast. It's hot if you've ever lived in Atlanta. But it's interesting, there's not a lot of West Nile going on in the Southeast of the U.S. I mean, there's cases, but nothing like you see in the Midwest or in California or Chicago area. And the reasons we don't know. Clearly there's more to it than just hot. And but it's a good question. We thought Florida would get wiped out, and it didn't. So if you figure it out, tell me. Because we don't really know the answer to that. They've got plenty of competent vectors there. Because it's that southern house mosquito. You saw the map went all the way across the southern U.S. So it's the same vectors.
So it's different than the mosquito in the west and the mosquito in the north.
Well, there's
Transmission different?
there are certainly really competent vectors in Florida, and so we don't really know why. St. Louis encephalitis, which is a related virus, has been a problem in the past in Florida. So we thought Florida is going to get really hard hit, and it didn't. And still a mystery, why not?
Maybe people in Florida use their bug spray the way they're supposed to.
Or never leave the air conditioning.
Well, they
Yeah.
do have a very active spraying program throughout the state there too, because of eastern climate also, so maybe they.
Right, it could be control efforts, but it really is in that whole southeastern U.S. you just don't find very high incidence of West Nile for reasons that really aren't entirely clear.
So sort of a follow-up question to that, also from our Twitter universe. So West Nile first emerged in New York and on the eastern seaboard, but it seems like since then we haven't really seen these big outbreaks in that area. Do you think it's something particular about the weather patterns there, or that maybe the birds are more immune in those areas and it's not able to outbreak as much?
There has been persistent outbreaks in the Northeast. Remember, the outbreak in 1999 in New York City only had about 50, I can't remember the number, but 53 or something like that, neuroinvasive disease cases. And in the last few, like last year, there was quite a few. I don't know the exact number. I think it's just the situation in the Northeast is just kind of overshadowed by these massive outbreaks like we saw in Texas.
Good talk. I missed a part of your answer to, I think somebody asked about the mosquito control, and probably part of your answer was that, for example, a place like Texas is so big or so large that maybe the methods that are used for that would be so sufficient. I was under the impression that similar methods were used for the control of mosquitoes during the malaria eradication process, and how would that compare with what you have? And the second part of my question is, you talked of IVIG as effective or showing signs or some promise, and you say that that is likely because maybe people from the Middle Eastern area had developed some cumulative antibodies against this virus. Most of the talk that I've read about of how IVIG works is through the anti-inflammatory aspect of it. Do you think that that is probably the problem, that a West Nile virus infection is more of an inflammatory problem, and therefore you need the anti-inflammatory?
I think it's probably both, because clearly in our very immunosuppressed patients and our transplant patients, they get very severe disease, and there's not a large inflammatory component. We can PCR virus that they have among the worst disease. So, but in an immunocompetent host, the inflammatory response definitely has a role. So, IVIG, we use it for many different illnesses, and we use it for both reasons. So it definitely has immunomodulatory properties, but it's also important for the actual neutralizing antibody that's present in the IVIG preparation. So for instance, if we have a bone marrow transplant patient with various respiratory diseases, we will use a product that has high titer to that particular virus to help clear the virus itself. But for other illnesses such as Guillain-Barré disease, which is a post-infectious, inflammatory demyelinative condition, we use it for the immunomodulatory properties. So it's both. Then I'll let you answer the first part.
Okay. The answer is to control mosquitoes, there's basically three possibilities. One is to get rid of breeding sites. So Culex mosquitoes are container breeding mosquitoes. They like to breed in small pools of water, like flower pots, etc. So strategy there is trying to eliminate breeding sites for Culex mosquitoes. Second thing is to do larviciding, to put in larvicide to kill the aquatic forms of the mosquitoes before they emerge as an adult. And so places like New York City do very active control, like putting larvicide in storm drains where water accumulates. So those strategies, you try and get rid of the immature forms of mosquitoes before they turn into adults. That always doesn't work, obviously, because we have these outbreaks. And so when an outbreak happens, the name of the game is to try and kill the adult mosquitoes that are already infected. And the way to do that is to spray pesticide using ultra-low volume spraying, where less than an ounce of pesticide is sprayed per acre. You actually need very, very little pesticide. And so you can either do that by spraying from a truck, going up and down the streets, which people don't mind so much, or spraying from an airplane, which people do mind a lot, even though it's the same stuff. It's just that people have a thing about airplanes flying over, spraying chemicals over them. But that's basically that. Those are the options. Now, to do that in a place like Dallas, Fort Worth area, you have a pretty big geographic area, a lot of people in it. And so it costs more than $2 million to spray that area. So really these mosquito control efforts are really only appropriate or used for urban population centers where you have a lot of people at risk in an area that's either highly endemic or an area that's about to experience an outbreak. Oh, the other thing you mentioned about malaria control. Malaria, it's a completely different thing because the malaria is spread by Anopheline mosquitoes. These are Culex mosquitoes. Anopheline mosquitoes are going to bite people in houses. That's why bed nets are good. These mosquitoes are primarily bird-biting mosquitoes. So they're going to bite you primarily outdoors.
We can take one more quick question and quick answer because it's 8 o'clock.
Dr. Peterson and Dr. DiBiase, thank you so much for the exciting presentation. You mentioned nucleic acid testing and serology. How often are blood donations screened?
You can speak to this better than me, but that is a standard test for our blood pool. And then there are larger epidemiologic studies that look at the prevalence of detectable virus in our blood pool.
Right. Since 2003, there's been universal screening of the U.S. blood supply, which has picked up thousands and thousands of positive donors. And you saw that pyramid that both Roberta and I presented. It's that bottom part of the pyramid with several million people infected who are asymptomatic that actually present the risk. Because these people feel fine, but they've got virus in them, so they donate blood or donate organs. And there is some transmission that goes on in the U.S. year-round in the very southern states, and that's why the blood supply is screened for the entire year.
I have a few thank yous to impart before we thank our speakers. This series, Microbes After Hours, is sponsored by the American Society for Microbiology. If you're not a member and you care about supporting the science of microbiology and publicizing the importance of microbiology in keeping our country safe and healthy. You might want to consider joining ASM. I also would like to thank our communications department that streams these evenings online, Chris Kandayan, and our cameraman. And this is a joint effort between the American Academy of Microbiology, which I direct, and the communications department. But of course, I direct the Academy, so I don't do any of the actual work. Shannon Green does all of the work to bring these meetings together. So I'd like to, in your applause, include those people, and especially to our two speakers tonight.